I'd like to do the draw for who wins the £100 gift voucher for completing the survey. So click on there. Okay. So if I now spin the wheel.
Oh, that's clever.
Oh, I say.
Oh, wow.
Isn't that good?
Jerry was, don't you? He died.
Who's 19?
It wasn't that entertaining. Number 19, I've looked at my list of respondents. Number 19 is R.S. Gregory, who I can't say that I know, but ring them and contact them and let them know there's a £100 gift voucher on their way. So,
Good.
which is brilliant. So I'm really chuffed about that. So sorry, Jordan. I hope you like the spinning wheel.
Yeah, it's a great one. That's a great thing. All right.
Let me put you on spotlight for everyone.
Okay.
If I ask everybody around the screen, please to mute yourselves. If I see that you're not unmute, if you haven't muted yourselves, I'll do it for you. You're up. Yep, you're on.
All right, thank you. So as you can see, I'm in my office. It's quite a busy office, so there will be a little bit of background noise, but I'll sort of suppress it with my voice, hopefully. So my name is George Gverich, and I'm the manager of Parkinson's UK Brain Bank at Imperial College. I'm just going to go through my presentation today and talk about the Brain Bank and how we're contributing to this huge area of research that's going on Parkinson's disease, where people are trying really to find out what's going on in Parkinson's and bring on better therapies and better treatment for people with Parkinson's. So I'm going to start sharing my screen now. Oh, can you actually enable me? Because
hold on, sorry. There you go.
All right. That would be this one here. I hope you can all see it now. Is that okay?
Yeah.
Right. So excellent. So this is this is quite an old presentation. Don't look at the the dates. So to start with, it's a little bit of kind of a overall view of what we can do and what we're doing. What we're trying to do, number one, is to fulfill the last wish of all those who are willing to support research in this rather specific way by donating their tissue after death. And then the other main important area of activity is to preserve this tissue and make it available to researchers. Apart from that, we also work with health professionals. We provide a bit of educational, we have an educational role there, sort of talking about Parkinson's and what's behind Parkinson's, and especially about the role of pathology, which I'm going to illustrate a little bit later on. And finally, we are one of those flagship facilities of the Parkinson's UK, one of the few that Parkinson's UK has been supporting for now almost 20 years, and we're part of their research strategy as well. If you have any questions in the meantime, just let me know. You can ask them, just unmute yourself and ask the question. I'm very happy to answer them and to stop the presentation. This is just an brief overview of history. So Parkinson's, as we know it, is not an old condition, although it has probably been in the human population for many, many years, but it's only been described in the 1800s by Dr. James Parkinson's, and it was named after this doctor, so that's why it's called Parkinson's disease. And the number of different people contributed to description of Parkinson's disease to the point where in 1950s a new drug was introduced, which is still around, and I don't know whether that's still a good thing or not, but we'll see a little bit more about it later. There are about 120,000 people in the UK who suffer from Parkinson's disease, and about 10,000 people, they say, in the UK are diagnosed, newly diagnosed each year. So the numbers are going up, and obviously there is an urge to find out what is going on in Parkinson's disease and why we're getting more people affected by this condition. So in order to talk about Parkinson's, we need to talk about a human nervous system and what it does for us. And this is probably the most important system in our body. And essentially you can say that that's what we are. We are the brain. And everything else is simply a packaging, and it allows brain to move around, do different things. The brain itself and the rest of the nervous system, they process information from the environment and the body itself, and that's how we gain this consciousness about what's going on inside of our body and what's going on outside of our body. This is all mostly done by nerve cells, and we have about 1 billion these cells. They produce the information. I don't know if you can see my pointer, but this is just a depiction of several nerve cells linked in a chain. And that's the main characteristic of the nervous system, is that these neuronal cells, nerve cells, they actually communicate with each other. And that's the most important part of the system, because that's how we actually convey the information from one part of the nervous system to another, and then finally to a target organ, whether it's a muscle or gut or anything like that. Nerve cells are very sensitive in many different ways, and they need a lot of support. And to do that, we have so-called glia cells or support cells. And these cells will support neuronal cells in many different ways. There are 3 main types of cells. One is astrocytes. These cells maintain the blood-brain barrier, which is very important because we don't want anything from the blood or anywhere else come into the brain. So the brain is very protected organ, and astrocytes are there to protect them by maintaining the so-called blood-brain barrier, the barrier between the brain and the blood vessels. Then we have cells called oligodendrocytes, which you might see here, and they produce this insulating layer around neuronal cell processes. And this allows neuronal cells to conduct electrical impulses very quickly. Without this insulation, these impulses would travel much slower, and then we would obviously experience some symptoms. This is more typical, for instance, for multiple sclerosis, which is known as demyelinating disease, because myelin is stripped off these processes called axons. And then there is a problem with variety of different functions in the body. So these are oligodendrocytes. And finally, we have a group of cells which are called microglia macrophages, and they're part of the innate immune system, and they're there to protect the brain or this whole central nervous system from viruses, from bacteria, or anything else that might harm the nervous system. So this diagram depicts the whole problem of Parkinson's disease, and the main issue is the lack of a chemical substance called dopamine, and this most of you probably know about it quite well, because that's the actual drug that's given to patients with Parkinson's, and it's a substitute drug, obviously a wonderful thing, but does not work all the time, and there are obviously problems associated with it. So what we're seeing here is a part of the brain which is called substantia nigra, and this part of the brain, and translation is essentially black substance. It contains neuronal cells which are pigmented, and that's why we can see them when we cut the brain and when we look at the slides. And these pigmented neurons, they contain dopamine. They're the main source of dopamine in the brain, apart from some other areas as well, and they're involved in regulating movement. And this is not really a simple thing, switch on and off, but substantia nigra communicates with other parts of the brain, which is basal ganglia here and thalamus, and then cerebral cortex, where the motor cortex is actually the main executor of movement in the body. And they essentially regulate between themselves whether movement is continuous, whether it will stop, slow down, speed up, and so on. Once you lose dopamine, then the regulatory mechanism is gone, and then whoever is suffering from this problem will start experiencing problem with movement, and that's why we call Parkinson's movement disorder. Obviously, this is just a part of the spectrum of Parkinson's. There are many other problems that are coming later on, but movement disorder is the first symptom that we see in people. So problem with walking, tremor, and so on and so forth. There are a couple of pathways which are regulating, which are just the ways that the brain is wired, and I'm not going to go into too much into these details unless you want more information. One is called direct pathway, and the other one is the indirect pathway, and one is actually facilitating the movement, and the other one is inhibiting the movement. If you don't have enough dopamine, then the balance between these two pathways is damaged, and that's why people experience problem with regulating their movement and initiating movement and so on. The main pathological substrate in Parkinson's is an accumulation of a protein which is called alpha-synuclein. And this alpha-synuclein, you can see in one of these pictures here, accumulates in neuronal cells. And the current theory is that alpha-synuclein is essentially damaging the neuronal cells and leading to their death eventually. And that's why you actually losing many of these factories, dopamine factories, and finally losing the ability to produce enough dopamine. Other problems that were identified was also accumulation of iron, calcium, and also inhibition of neuromelanin, which is the pigment in dopaminergic neurons. So all of these are still at the level of kind of advanced hypotheses, and we really don't know what is causing Parkinson's disease. The drugs that, there is a whole spectrum of drugs that are available to someone with Parkinson's today, and the aim of this therapy is to increase the level of dopamine, because that's what's missing in Parkinson's. Also to stimulate parts of the brains, the cells that can still produce dopamine, to produce more of it, and also to prolong the life of dopamine in the brain by blocking the action of variety of chemicals, enzymes, and so on, that may affect the dopamine in a number of different ways, from inhibiting it, simply stopping it from binding to targets, to receptors, or making sure that dopamine is broken down too quickly or too slowly, and so on. So levodopa is obviously the replacement therapy. That's the actual dopamine. And it is essentially, it converts into dopamine in the body using a number of different enzymes that help this happen. And then once that's not enough, which is a few years into Parkinson's disease, then neurologists will start adding another set of drugs. Some will be, one of those are called dopamine agonists, and they essentially work together with dopamine to enhance the effect of dopamine, or enhance the effect of dopamine itself on neuronal cells, and therefore ease the problems with movement. Later on, there are a number of so-called inhibitors used, and these are there either to inhibit enzymes that chop the dopamine and sort of inhibit it, or also to inhibit the inhibitors of dopamine. It all gets a little bit muddled after that. And there are a number of other drugs that are actually supporting different parts of dopaminergic pathway, and therefore, you know, supporting the movement in some way, and so on. The problem with all these drugs are that they are not that selective, and the brain is a very complex place to treat, especially because, number one, not all of drugs will cross this very regulated blood-brain barrier, and number two, they would essentially act on many different areas in the brain, some of these that actually don't need any action. And this will give you a lot of side effects, and these can be, you know, varying from a sort of a minor stuff to major psychosis, hallucinations, and so on. So the race is on really to devise a new drug, which, number one, can be delivered straight into the brain, and that's what Parkinson's UK tried in one of their recent trials with growth factors. You might have seen it on BBC at some point. And this was a real breakthrough because this drug was delivered straight into the brain and into an area where it was expected to work best without affecting the rest of the brain. So there was a mixed success with it, but at least what it has shown is that we have the technical ability to deliver drugs to the brain and to the place where the damage is. And then we're also looking at a number of other different drugs that can actually attack whatever is causing Parkinson's disease. And I'm going to say a little bit more about it later on. So this is the new type of therapies that are proposed for not just Parkinson's, but many, many other conditions. It shows a lot of promise. Gene therapy is probably going to be used reasonably soon. And this is essentially to change the faulty genes or introduce a new gene that can provide some kind of product that will support, for instance, neuronal cell survival in Parkinson's disease. It can produce these so-called neurotrophic factors. These are chemicals that support neurons in surviving, in their function, and in resisting any kind of damage. So there's a great hope that gene therapy might deliver this, as I said, reasonably soon. And then we have a stem cell therapy. Probably you've heard a lot about it already. Essentially introducing these new cells, which are unspecialized cells. They can divide and they can produce absolutely any type of cell. And the hope is that we can deliver these cells into an area where certain type of cell is missing, such as substantia nigra, or where the dopaminergic neurons are dying. And then we can promote the differentiation of these cells into new dopaminergic neurons and therefore resolve the problem in Parkinson's. There are a number of problems with essentially delivering these cells and also making them differentiate or change into the desired cell type. So this will probably take a little while before we actually can use them in the brain, maybe in the peripheral organs at some point, but not so much in the brain for a while. So the brain back itself, to go back to what we actually do now, is running a UK-wide donor scheme, and that's one of our main activities. And our part of this scheme is community-based, and that means that we are getting donors with all types of Parkinson's. There's a huge range and variety among Parkinson's patients, and this approach allows us to collect tissue from a variety of people with Parkinson's and to have insight into variety of different pathologies. That's very important, because then if you're focusing only on one thing, you are actually skewing the whole research, and then you're sort of neglecting a part of the population which is suffering from a different type of Parkinson's disease with a maybe even different cause and so on. However, we're also interacting with a few major UK patient cohorts, and three of them are listed here. So one is Oxford Discovery, which is still ongoing. Pine, that has been discontinued a couple of years ago, and Proband is in its final phase, and they will be closing soon. So we have approached these cohorts, and we talk to their patients, and we ask them if they would like to join the donor scheme, because this way we're not only getting a lot of very, very good, very thorough clinical data, but we can also support it with very good pathology. And you'll see why is this so important for trials like this to actually succeed. In order to register on our donor scheme, one will have to request a donor pack, and which consists of several forms which need to be filled in just to give us a permission to actually collect this tissue after death and to collect medical records after that as well. And this is the content of this donor pack. And this is what the card looks like. It's actually changed color now a little bit, and the number is different, but Nevertheless, it's one of those credit card donor cards which you can put in your wallet and carry with you if you want, and then there is a number to remind you who to call and when to call. And then when the question arises, who can donate tissue to this particular brain bank, it's absolutely anyone. We do specialize in Parkinson's disease, but also Parkinson's plus movement disorders. So those are sort of related conditions. And among those, these are multiple system atrophy, progressive supranuclear palsy. There's also corticobasal degeneration, frontotemporal degeneration, and so on. There are certain limitations. If you have another major pathology in the brain, then the whole pathology, whole picture becomes very complex. And in some occasions, if someone has a massive stroke, or if there is a metastatic or primary brain tumor, Creutzfeldt-Jakob disease or systemic infections, we unfortunately have to reject the donation. And because it's either too complex to do research on, or there is a health and safety risk, such as in the case of CJD and systemic infections, those antibiotic-resistant infections. And one of the new ones now is COVID, although we can deal with COVID in different ways. So we still receive brains with COVID. And there were some quite interesting findings that we've seen now in the brain and also in the body, the recent study that we've done. In addition, we need controls. So people who don't have any kind of neurological condition or even different neurological condition. So they're important because this is a relatively healthy tissue. I mean, when someone dies, there are certain changes in the brain. So you never get a sort of what you would call, let's say, normal brain or whatever it is. But it is very important to have something that will be used to establish a threshold in any type of research project. So you would use a control to work out whether a certain, for instance, gene or a protein of interest is actually there in the sort of healthy human brain, and what sort of amounts we can detect in a healthy human brain, and whether this goes up or down in Parkinson's disease. So that's why controls are important to compare with what's going on in Parkinson's disease, and to give us an idea whether what we're looking at is essentially relevant for someone with Parkinson's. So when the time comes, the brain bank takes over the whole organization of tissue donation. We are only dependent on health professionals and relatives to let us know about the death. And then we will take over the whole organization. So we will contact the GP to inquire about the death certificate, which is very important because these days we just can't do anything without the death certificate. We will do all the admin stuff, make all the mortuary arrangements, and transport the tissue finally to the lab, and then start preparing tissue and preserving it for all the future research projects. Tissue preparation involves first inspecting the brain on the outside. That's probably going to be subject if you come to see the brain bank in person, and then I can show you what we're looking for. But then looking at, then we need to essentially cut the brain, slice it. And you can see on this side here that we start here in the middle of the brain. This red line depicts the first cut, and this is essentially necessary to expose the substantia nigra. And down here you see 4 images. On this side is a relatively normal substantia nigra. So you can see this dark line here. This is where all these pigmented neurons are crammed with neuromelanin and dopamine. So these are dopaminergic neurons. And on this side, you can see that there is still pigmentation, but there's a much less of it compared to the other image I've shown you. And then same on this side here, substantia nigra is virtually non-existent, and here is very pale as well. So pale nigra essentially let us know that there is something going on in this part of the brain, and we may start suspecting Parkinson's at this stage. So this is only a first indication that someone might have Parkinson's, because there are other conditions that can affect substantia nigra. The damage to nigra starts from the side of the brainstem, and you can see here how the pigmentation is lost here, but there is some still remaining towards the middle of the brainstem, which is this is the middle line. And here it's essentially all gone. There's a little bit of indication there is some pigmentation there, but that's about it. The characteristics of nigra is that these neurons are basically being lost throughout your lifetime, but the degree that by which people without Parkinson's, people with Parkinson's lose neurons in substantia nigra is different. So in normal circumstances, people will be losing something like 0.9% of cells in this part of the brain, whereas with Parkinson's, it's estimated that loss is more than 1%, something like 1.1, 1.2. So that means that if you have 100 cells, healthy brain will lose one cell.
Hi, not too bad.
All right. So the healthy brain will lose about one cell, and whereas someone with Parkinson's would lose actually 0.1 cell, and someone with Parkinson's will use more than one cell. And that essentially accumulates over time. It goes over a long period of time, probably some 10, even 20 years before someone experiences symptoms of Parkinson's disease. So by the time there is a significant loss, I think more than 50 or 60% of these cells, then will someone start experiencing usually motor symptoms to start with, and then later on some other symptoms such as dementia and so on. So once we slice the brain, we'll place it on a grid like this, and then we'll cut along these grid lines to generate tissue blocks, which will be in some cases frozen, and in some cases these blocks will be fixed in a chemical called formalin. The best way to preserve brain tissue is to simply freeze it, which means that we are immobilizing the water molecules, which might activate the remaining enzymes in the brain or the contaminating substances, bacteria and whatever contaminates brain after death. And this way we're preventing brain damage. It's a very soft tissue. It's filled with blood. There is a lot of there are conditions which will favor degradation of the brain, and it's a very quick process. So therefore we need to act very quickly, and the time we have is something between 24 and 48 hours. Brain needs to be preserved, and the freezing is the best way to do it, because tissue preserved by freezing is suitable for all known experiments that we have these days. Tissue that is frozen will be stored in these boxes and then put in these deep freezers. The temperature is -80. These are airtight containers, and the freezer banks are secured, obviously with controlled access. And the coding system is called linked anonymization, which means that only few of us actually know the link between the case number, which is assigned to the donation, and the actual identity of that donor. So researchers will never find out the identity of the donor. Data are always protected. All the identifiable data are protected. Very importantly, the system that we're using, this grid system, allows us to pinpoint different anatomical areas in the brain, which might be of interest for Parkinson's research. And for instance, on this picture, if you look here at this coordinate D2, there is a slightly convoluted area, which is it's a brain fold called hippocampus. And this brain fold, hippocampus, is functionally responsible for short-term memories. And this is one of those areas that is affected in Alzheimer's disease. But it's also a place where endogenous, so internal brain stem cells are stored. So a lot of researchers are interested in order to look at hippocampus, number one, from the point of dementia and problems with memory, but also from the point of researching the stem cell potential for replacing damaged parts of the brain. So if they were trying to look whether we can mobilize stem cells from the hippocampus here and actually repopulate some parts like substantia nigra to produce new dopaminergic neurons. So this grid system essentially allows us to pinpoint this when the blocks are cut and stored. We know that our D2 is essentially hippocampus, and we can retrieve it and pass it on to researchers. So as I said, frozen tissue is very, very useful. It's a high quality, when we say high quality tissue, it's sort of a lifelike. It's something, you know, that people would have in their brain. So all the proteins, all the genes are more or less unchanged. And it can be used in any experimental setup. And nowadays, we are also isolating cells from these brains. These are live cells. We grow them in culture. The important thing with these cells that they can be propagating over a long period of time, and they can be turned into stem cells. And therefore, we can look at these stem cells, or we can look at, we can generate neurons from these stem cells, and we can look in the dish what is actually going on with Parkinson's neurons and compare it to control neurons. So that gives us a more insight into what might be going on in Parkinson's, and are there any functional defects in neuronal cells in Parkinson's. And these are also important for those so-called omics studies. These are what they call genomics, proteomics, and so on, very popular these days. These studies have changed the science recently, because in the past we were only able to look at one gene, maybe two, or protein at the time. And with genomics and proteomics and omics in general, we can look at number of different data points. So we can look, for instance, the 100,000 genes on the chip. We can look for thousands of proteins and look at the same time at all of them and get these data out of these studies and see whether there are changes in particular proteins and how they actually affect each other, how these proteins that are changed affect other proteins, and similar for genes. As I said, unfixed tissue, that's before we actually do any preservation, can be used for cell isolation and generation of these cell lines that can be studied further down the line for variety of different changes. And then we have so-called fixed tissue, where we use formalin, very, very old, very traditional way of preserving tissue. We do this when certain conditions are not favorable, or we cannot reach the mortuary in good time, and some other conditions were not good enough. And this tissue is fixed. Now, this is still very important. Number one, this is preferred for pathology and also for imaging studies, because fixed brains can be imaged in an MRI scanner, for instance. And then we can do the pathology on a variety of these parts of this brain wherever MRI shows some changes. And we can feed this information back to the MRI people and improve the imaging itself. This is also our archival collection. We have brains that were collected some 50, 60, 70 years ago, and all sort of these periods in between. And this allows us to see whether anything is changing with Parkinson's disease. We haven't seen anything so far, and not much has actually changed with Parkinson's, because as I said, since 1950s, the therapy has stayed the same more or less. And nothing else has happened in Parkinson's since then. So we haven't seen anything. But still, fixed tissue again is here, and we can always look back and see if there's anything that we might have missed in those very old brains where there was no actually therapy, there was no dopamine and nothing like that. So once we get to the point where the tissue is preserved, the next stage is to characterize the tissue and to diagnose it properly. And you will see that clinical diagnosis differs from the pathological diagnosis. The real value of looking at brain sections is that, as someone said it looks like a crime scene. And essentially, all the evidence of a crime is there. Anything that might be causing Parkinson's will leave some trace in that tissue. Our main problem is to actually unravel this evidence, to find this evidence, and then use it to build our case for essentially accusing something or some agent or someone of causing Parkinson's disease. In order to do that, we use each one of these blocks that I've showed you previously is cut into very thin sections, and then these sections are stained with variety of probes which tell us about different aspects of pathology. So we can use a probe that detects alpha-synuclein in these sections, and that will tell us about the spread of Parkinson's disease. We can use two probes, one for beta amyloid and tau, to show us the spread of Alzheimer's pathology, which quite often comes with Parkinson's disease, and some special stains to show some other pathology that is usually associated with aging and so on and so forth. So once we look at the medical records, we will probably come across a number of clinical diagnoses. One very frequent, obviously, is idiopathic Parkinson's disease. So that's the standard Parkinson's disease. Idiopathic means unknown cause. And this majority of our donors are coming with this particular diagnosis. Actually, all of them come with this particular diagnosis. Smaller number of people comes with the diagnosis of MSA and PSP, or some people with dementia with Lewy bodies, which is essentially pathologically identical to Parkinson's disease. The only difference is that dementia occurs very early during the course of what looks like Parkinson's. And finally, there are cases of vascular Parkinson's. This is down to problems with blood vessels, to a lot of mini or big strokes, inflammation of blood vessels, atherosclerosis, and so on. So this is not your typical Parkinson's disease. There is no accumulation of alpha-synuclein. There is no damage to nigra, but still there's a lot of damage to the brain itself, mostly due to hypoxia and due to obviously problems getting oxygen into the brain and getting glucose, so food into the brain, and therefore some people develop Parkinson's symptoms. Neuropathological diagnosis, on the other hand, is focusing on how much this pathology has spread within the brain. Number one task is to confirm the diagnosis, and we can do that in 85% of cases. So clinical diagnosis is okay in about 85% of cases. What we do is we use this so-called Braak staging, and this Braak staging essentially tells us where the pathology is. So if you're looking, there's a Braak 1 to 6. The Braak staging 1 to 2 is early Parkinson's, usually incidental Parkinson's, something that we see in our control donors. And this is mostly pre-symptomatic, so there won't be any kind of symptoms, motor or any psychological cognitive problems. Braak stages 3 to 4 is an established Parkinson's intermediate stage, and usually characterized by the motor problem. So this is a motor phase of Parkinson's. And finally, Braak 5 and 6 is the late Parkinson's, and usually goes together with dementia. This essentially describes where we're seeing pathology in Parkinson's disease. So the early Parkinson's is mostly in the brain stem. The established one spreads towards the sort of deep brain centers, which is called basal ganglia, amygdala, hippocampus, one of those as well that you've seen on some previous slides. And finally, this is when the whole cortex, so the gray matter of the brain, is affected, and there are a variety of problems which cause cognitive problems to people who suffer with this stage of Parkinson's. The other problem with Parkinson's is the age that at which people start experiencing these problems. It's usually between the age of 60 and 70, or 70 to 80 even, apart from those rare cases of early onset Parkinson's disease. And therefore, the older population starts already having changes in the brain that are, to some part, they're part of normal aging, but there's some of these are part of so-called pathological aging. And Alzheimer's disease is one of those pathologies that we see in pathological aging. And this can be present to various degrees, even to the point where people have both Alzheimer's and Parkinson's at the same level. It's just we're using the same Braak staging, so it'll be Braak 6 for Alzheimer's and Braak 6 for Parkinson's. Apart from that, we will find a number of problems with blood vessels. There will be, which are usually associated with aging, but also with pathology within blood vessels. We're going to find a number of different neurodegenerative conditions that might be associated with aging, or might be just there because there's something else going on, and so on. So diagnosing Parkinson's clinically is obviously not an easy task, and most of the people are essentially underdiagnosed because we don't account for their Alzheimer's, for problems with blood vessels, and so on. And some people are unfortunately misdiagnosed. So the importance of pathology. So I'm going to show you just a few summaries, pathological summaries, findings from several of our donors. So donor number 1 came to us as an idiopathic Parkinson's, with idiopathic Parkinson's diagnosis, and neuropathological diagnosis was advanced Parkinson's, so that's what we call BRAC6, and also early Alzheimer's, which would be a sort of BRAC1 or 2 maybe. Pathologists tend to call Parkinson's Lewy body disease because Lewy bodies are the main pathological features. Those are essentially, they look like orange blobs, which are filled with this protein, alpha-synuclein, and they're very characteristic of Parkinson's disease. Donor 2 also came to us as idiopathic Parkinson's. So this was the final diagnosis and the working diagnosis, and this person obviously was receiving therapy for this diagnosis. The neuropathological diagnosis is something I already mentioned, that was mixed dementia. So it was advanced Parkinson's combined with advanced Alzheimer's. So obviously there would have been a lot of dementia, different problems, but also different pathology completely. And I doubt that the actual therapy would have been different that much, because Alzheimer's doesn't really have a lot of options these days. But in any case, you know, this was only a partial diagnosis that compared to what we've seen down the microscope. Oh, sorry. So the donor number 3 also came, again, these are all idiopathic Parkinson's or classic Parkinson's disease. The neuropathological diagnosis, surprisingly, was a control brain. So no Parkinson's and very early Alzheimer changes. So the pathologist mentioned possible genetic mutations, mutation of one of these genes that might be causing some Parkinson's symptoms, but not really a typical pathology of Parkinson's disease. And finally, we had a case again with Parkinson's. However, it turned out to be Creutzfeldt-Jakob disease or mad cow disease, which is a completely different condition and something that creates a lot of problems for anyone involved, and also early Alzheimer's at this stage. So this is the whole range of donors and whole range of diagnoses that we might encounter during this work. And knowing that we receive 120 cases every year, we can say that usually we can split these cases into something like 5 different cohorts, depending on what we find pathologically, and all of them come to us diagnosed with just Parkinson's disease. So move on with our research side. It's essentially, we are here to support the worldwide effort to unravel what's going on in Parkinson's disease. And we had quite a bit of success with that. So you can see on this map all the countries that we reached so far. And I can freely say that anyone who's doing anything in Parkinson's disease has at some point approached the brain bank for tissue in order to do some of these experiments. So it's very important that this sort of facility exists, and that we actually have tissue to provide for this research, because Parkinson's disease is a very much a human condition, and it's important to look into this into human brain at least at some point during any kind of research project, because you really need to account for what's going on in a human brain to establish whether whatever you're doing in some animal models or in a dish or whatever is essentially relevant to someone with Parkinson's disease. And the aim is obviously to, number one, see what's going on in Parkinson's, why these cells are dying, to find new drugs to treat Parkinson's, to identify genes that make someone susceptible to developing Parkinson's disease and therefore prevent Parkinson's, hopefully, in the future. So as I said, supporting science, a number of these different studies are available to scientists these days, whether they're in vitro, basically in a dish, in vivo, so this using cells and animals, And now these new ones in silico studies using only computers to just crunch the data. And we also need obviously a lot of data for this. That's why it's probably worth mentioning that we are trying to set up something called Parkinsons Register, where we would like people with Parkinson's to donate their data, to do some cognitive tests, do some surveys, in order to establish really their individual experiences and individual circumstances relating to their Parkinson's disease, and to somehow put this all together and see if we can find any new leads in kind of trying to explain what's going on in Parkinson's. We're trying to support what they call translational research or target validation. Now, that means simply translating results from another research system into a human system. Is it relevant for someone with Parkinson's? Simple as that. And also to target validation means, you know, if you found a gene in a mouse or a rat or whatever model you're using that might be changing something in your model of Parkinson's disease, does this gene actually exist in a human in the same form, and can we use it to modify Parkinson's in humans, in patients with Parkinson's? And also we tend to provide also a lot of feedback to absolutely everyone. We collect all the data from the researchers for every single sample they used, and we will provide it to new research projects, but we also provide data to relatives and health professionals who looked after our donors, and usually in the form of a neuropath report and a clinical summary and so on. So this is just, I'm just going to go quickly through these. These are just different ways of looking, why we're looking at cell death in Parkinson's disease. That's the main problem, is this accumulation of this protein, alpha-synuclein, and also subsequent death of neuronal cells exposed to alpha-synuclein. There are a variety of hypotheses why someone would get Parkinson's disease, or how whatever is causing Parkinson's disease gets into the brain. And there are some very interesting developments these days, and that's why Brain Bank has changed the focus a little bit. So nowadays we're also collecting, apart from brain and spinal cord and all the parts of the central nervous system, we're also trying to collect samples of gut tissue whenever we can. It's not an easy thing to do, but it's very important to start collecting this. The current estimate is that approximately 40% of people with Parkinson's, their disease starts in the gut, not in the brain. And there were some very interesting studies quite a few years ago when people with the peptic ulcer, so they were treated with a surgical method called vagotomy. So there was actually, surgeons would cut the vagus nerve, and that's the very important nerve that connects the brain stem directly with the gut, because brain really needs to have a control of the gut, constant control, and it receives a lot of feedback from the gut. So you would simply cut this vagus nerve, and then the people who were experiencing peptic ulcers or stomach ulcer, they would sort of recover to some extent. However, cutting this nerve would cause a lot of other problems to these people, problem with gut motility and so on and so forth. So they've changed the method into just partial resection of the vagus nerve. So they would cut the fibers that go to the gut, to the stomach, but not those that go to the gut itself. And what they've seen is then when you do a total vagotomy, so cut the nerve completely, partial vagotomy and intact vagus nerve, they would actually go on and develop, in some circumstances, Parkinson's disease. And later on, research showed that a variety of events in the gut can, to some extent, in some way, promote Parkinson's changes in the brain. And the main one at the moment that people are focusing on is the gut inflammation. So inflammation in the gut, which can be related to changes in the flora of the gut, so bacterial content of the gut, can be related to anything that is in the food. It could be any kind of chemical that's additive or added to food for anything like that, and so on. So, but nevertheless, now we know that at least in 40% of cases, whatever is going on in the brain will start first in the gut and then propagate into the brain, usually via the vagus nerve or via the spinal cord. That's still undetermined. The other way that brain can be affected is via what we call olfactory pathway. And you probably know that people with Parkinson's tend to lose sense of smell quite early into their condition. The part of the brain that actually picks up all these molecules in the air is called olfactory system, and contains of two little protrusions on the base of the brain, which end up just above the nose. And this is the part of the brain that's most exposed to the environment. So therefore, the hypothesis is there's something in the environment where there could be some chemicals. Usually we accuse organic solvents, anything like that. They might be damaging this olfactory pathway and actually precipitating all the pathological changes in the rest of the brain. So these are the two ways where someone might get Parkinson's disease, which then causes death to these neuronal cells. The culprits could be anything like neurotoxic components, something that's toxic to neurons. These are free radicals. You probably hear about them. There's when you're using sort of antioxidants and that sort of thing, that's what they do. These are some oxygen chemicals that they have a high reactivity, and they can damage cell components and therefore cause cell death. Accumulation of iron in certain forms, or any kind of toxins from the environment. And then the unexplained role of the Lewy body, which I've mentioned already, lack of deficiency of systems in the body, such as antioxidants. Then we are still looking at the role of inflammation in Parkinson's disease, and how this damage spreads, which I've mentioned, could be from the gut, it can be via olfactory bulbs, and so on. The other way we're trying to unravel Parkinson's by looking at all the genes in someone's genome, and trying to find out which of these might be relevant for Parkinson's disease. And this is, for instance, one of those chips that can have something like 60,000, 100,000 genes. And we're getting now 100,000 different data points. And we're looking at essentially whether these genes are what they call up-regulated, so more active, or down-regulated, less active. And we're looking at particular cells, just which I'm going to show you now on the next one. So we can use what we call laser capture microdissection, and you're looking at one neuronal cell here. And using the laser, we can cut out this neuronal cell, and we can test it for a variety of different genes. So if this is a dopaminergic neuron, we can find genetic profile of a dopaminergic neuron from someone with Parkinson's and someone who does not have a Parkinson's, and then look at the data and see if there's any difference. So these are the genomics, proteomics, and now we're also looking at connectomics, which tells us the overall connections between brain cells within someone's head. And finally, if you ever have a wish to see the brains for yourself, sort of live tour, we have these visits, and we're very happy to organize them for you. So at some point in the future, once all this COVID stuff is gone, then just let me know, and we'll do our best to make it enjoyable for you. And that's all I had to say. If there are any questions, I'll be happy to answer them for you.
George, wow. Thank you. Absolutely wow. As Caroline just said, she understands more about Parkinson's than ever before. I thought it was
okay.
a wonderful, fascinating insight into an incredibly complex subject. And you've described it in layman's terms. I think all of us have come away with so much more knowledge. So for that, I think we're all very, very grateful, as we are for your time and sharing your knowledge. So yeah, thank you so much. If I take you off spotlight, if I may, and then ask back to gallery, hopefully you can click on gallery, everybody, and sort of see everybody on the call. Does anybody have any questions for our Pam?
Thank you very much. A most interesting and clear description. Thank you. My father's brain went in 2002 to the brain bank, and I'm hoping in time that my husband and ours will as well, as control and otherwise. However, would that brain bank have been the one that you're talking about, or was it an earlier concept of it, a general one?
It could be a few things actually. There is a brain bank. This particular brain bank moved from Institute of Neurology, Queen Square, in 2002 to Imperial College. So then we started as the Parkinson's UK Brain Bank. So we're essentially the official Parkinson's Brain Bank at the moment. But the other brain bank kept all the people who already signed up on their donor scheme. So that's the Queen Square Brain Bank. And then also there is a brain bank in Newcastle, which collects a variety of tissue samples, and among them also some Parkinson's tissue as well. It was because pathologists were quite interested in Parkinson's over the years. They've created a lot of smaller collections throughout the UK and also in the world. But as far as we know, we are now one of the biggest. We are the biggest brain bank dedicated solely to Parkinson's disease.
This was in 2002. My father had developed Parkinson's. I knew nothing of it until at the following day after his death. The hospital asked, would we be happy to donate his brain? So, well, that'd be interesting to know. But thank you. Well,
if you want me, I mean, if you just send me his details, we can check whether he's with us. Or,
no, that's all right. No, it's not.
That's fine. Okay.
Thank you. You've got more important things to do.
All right, that's good.
Deb, I see you've signalled putting your hand up.
Yes, I've actually got a few questions, but I don't want to hog all the time. So
that's all right.
The first question is, do you actually have enough brains donated?
That's a good question. That's a very good question. So I think I showed you substantia nigra, that tiny blackish line in the brain, and that's the most sort of part of the brain in Parkinson's research. And we can stretch it if we're really very, very careful to about 2 to 3 projects. And absolutely, I haven't seen a single Parkinson's project that hasn't asked me to provide substantia nigra for it. So on one side, we have a lot of tissue. On the other side, we don't have enough, never have enough. So it's a constant race of providing enough substantia nigra, because that's a reference tissue, and people need it to essentially establish really whether they're on the right track. Just like the control tissue gives you a kind of an idea whether there's something wrong in the Parkinson's tissue. Substantia nigra tells you whether you are on the right track as well. So we do need more tissue, because despite collecting the whole brain, the Parkinson's itself is focusing on a very few areas of the brain, and which are usually small in size, and so you do need to collect all the time.
Am I allowed some more questions?
Yeah, yes, okay.
Yes, one of the studies that my dad was taking part in was the presence of Helicobacter,
right?
Which I think was University of Nottingham. Are you actually linked with them and their research?
No, really, no. I don't know when was this study. That was that's probably looking if it was related to Parkinson's, they was probably looking at the role of the gut in Parkinson's disease, and Helicobacter is the one that's now a main culprit of stomach ulcers. So you need to treat Helicobacter first to treat, and then you've kind of sorted out the stomach ulcer. But as I said, any kind of inflammation in the gut can precipitate Parkinson's disease in someone's brain. So that, I'm guessing, that might have been that sort of study. It would have been really a great idea to link with these studies wherever we can. However, there are some ethical issues, and there are sort of their own local issues where they're not willing to collaborate with us. And also, brain donation is it's a difficult thing to discuss at if you're attending a trial for a drug that's supposed to make you better. And someone asks you, would you like to donate the brain when you die? So you really don't want to think about it at that time. I'm here really to find out about this new wonderful drug, and am I going to get better? Not really when I'm going to die, when I give you my brain. but yeah, we do approach those discussions at some point with suitable studies as well. So yeah, when we can, we do it.
Okay. And are you doing any research to any links with development of Parkinson's and head injuries, like they are doing with dementia and motor neurone disease?
We do. It's not, the brain bank is essentially just a facility for research, as we don't do any research per se, but we have a local research group who's interested in brain trauma and looking at essentially we have a largest collection of brains from boxers, British boxers from the 19th century. And these brains are still used to establish what's going on in the brain trauma and who actually gets this kind of damage. It's very, it's a new type of research. So far, we know that obviously, you know, there is no good protection from the brain. The best protection is not to hit your head and not to damage your brain. And, you know, all this protective gear for boxers and anyone else does very little anyway. So that's well established, but we also know that not everyone, even exposed to repeated brain damage, not everyone will develop this type of dementia linked to brain damage and Parkinson's disease. So only some people are susceptible. So that's the main aspect of this research, to find out what is going on and who actually gets this kind of damage in the brain.
Thank you.
My last question, if I could just have one last one. Was, are you actually doing anything about getting a mortuary yourself? Because one of the difficulties we had with my dad was trying to find, and I know it was in COVID times, but was trying to find somewhere that could actually remove his brain and spinal cord within the very tight time frame that you need to do it in order for it to be useful.
Yeah, we're trying. That's all I can say, I guess. Unfortunately, we, let's say it this way, we are the only UK-wide brain bank, and we're using every facility in the UK. So wherever our donors come from, we will try to use the local facility, local mortuary, to retrieve the tissue. That's the quickest, best, and fastest way. As well, fastest way already said that. Unfortunately, they don't always have the availability. Sometimes they're just not willing to help. Sometimes there are local regulations which stop us from doing it. For instance, now we have COVID, which has complicated a lot of things. And, you know, you can start with mortuaries that are absolutely fine with COVID, to those that don't want to hear about it, to those in the middle that will ask you to provide a recent test or to test the deceased, and so on and so forth. So there are always different things. We have discussed having our own mortuary, which is something, for instance, a Dutch brain bank has, but then again, they're in a very small country, and it's very easy to reach to, you know, different parts of the country to kind of access the donor. So at the moment, we're still just like limited to using all the NHS mortuaries. I have to say they're very helpful, but some days just things just don't go well, and we can't do it, and that's really unfortunate. But most of the times we're successful in getting the donation done. There are some ideas. Obviously, we also depend on the regulatory body, which is Human Tissue Authority. And they provide licenses to mortuaries to be able to deal with this kind of donations. And not every mortuary has a license. They don't want to apply for it. Might be too expensive, might be too much hassle or something like that. But they also trying to make it easier as well for us, as a Human Tissue Authority is now trying to license funeral directors to do brain retrieval for research. So hopefully that will be a really big change for us, because in that case, you know, we could make a contract with a local funeral director who is licensed, and we can use them to actually access the body and then do the tissue retrieval. So there is a bit of a hope that this problem will go away eventually.
Thank you, Deb. Thanks for some super questions. David, I saw you with your hand up.
Yeah, I was interested in what you said about the vagotomy.
Right.
How have we established that people who've had that can't get Parkinson's? I mean, is that just a matter of who didn't happen? And has anybody thought about investigating doing it and not having to avoid the problems of a vagotomy and avoid Parkinson's as well? Has anybody done that?
I'll go from the last question. It's doing a vagotomy. It's a very difficult to essentially to change it, because it's a very crude method. Essentially, you just cut the nerve and that's about it. So the best they can do is partial vagotomy, but that was done only for very specific reasons, and it's been completely abandoned these days. Now, it did lead to finding, it was based on sort of retrospective studies where they looked at who might be developing Parkinson's and looking mostly sort of statistics of Parkinson's, how many people should develop Parkinson's. And that's where they saw this anomaly that absolutely no one had Parkinson's disease, and that they concluded that something might be going on. And they looked then at the partial vagotomy and then discovered that, you know, some people do get Parkinson's as expected in these cases. So this is how they came up with this idea that something in the gut might be going on. And that then exploded, and then all these estimates were made, like what we have today, that 40% of people, Parkinson starts in the gut, although there is no clear explanation. Gut is essentially is a second brain. People tend to call it that these days, and it seems to be having a mind of its own, and also obviously talking to the brain. And it has all these links. What we're seeing these days is, it might be again another statistical study in the future. We used a lot of tissue from people who died of COVID, and we looked at where the COVID ends in the body. And we were specifically interested because there is this problem with smell in COVID, which kind of can linger on for many, many months and so on, so long COVID and so on. And it was quite obvious that people with COVID have some neurological symptoms, so it therefore affects the brain. So anyone interested in Parkinson's immediately said, like, why we should really see where the COVID is, hoping that we can explain something within Parkinson's disease. But the study shown there's very little of it in the brain. Even there's not a lot of it in the lungs as well, of those people really who died very severe COVID infection. But a lot of COVID actually ends up in the gut, in these what they call mesenteric system, so mesenteric nerves. And therefore this might create a substrate for people to develop Parkinson's later on. But this is something that we will see in the future, so we will follow that up. In which case, I'm not sure how much it will contribute to what we know about Parkinson's disease, but it will reinforce this link between the gut and the brain in Parkinson's disease. So hopefully, it will give us some new ideas that, you know, and we can then look how this virus is damaging the gut neuronal system, and how is this damage propagated to the brain itself, and how long it takes for this damage to go from the gut to the brain. So that's a new sort of a, you know, intriguing development linked with COVID. But, you know, it might tell us a little bit more about Parkinson's as well in a few years' time.
Thank you very much. I'm just looking at the clock, and it's nearly 5 to 12, and a number of us have got a committee meeting in about 5 minutes' time, and we want a quick loo break. So can I just ask people, can you put your hands together, please, for George and Via? I feel very privileged. I feel that we're a very privileged group to have had so much information and such a brilliant insight into what you're doing. I wish you every success with what you do, and, you know, please try and find a source for us. Caroline, one final question.
Will we have a link
Yes.
to how to get a package for donating?
Yes, I think Wille can circulate that if you don't have it already.
I can grab that off the website.
And to advertise it.
Yes.
Yeah, we'll be glad if you can do that. Thank you very much.
I'm sure David will do that in the newsletter.
Yeah, we will. Jill, before you go, yes.
Yes, very quickly. I mean, have you got any connection with the Cambridge Brain Bank? Because that's where I donated mine.
Ah, right. Yes, we're all part of the brain banking network.
Yeah.
So we all linked in some way, but not really direct connection. They collect info themselves. So we won't have sort of any insight in what they're doing, to be honest. But yeah, we're linked in some ways. We have this umbrella organization. So yeah.
But that was incredibly easy. My husband died in the early evening on the Wednesday, and I left a message, and they rang me back by lunchtime on Thursday to say, right, we've got it, thank you very much.
Yeah, that's the beauty of a local brain bank, because they have links with a local mortuary, and we unfortunately don't have that.
But,
right. Thank you all so very much. I think all of us around the screen have learned so much.
All right.
I wish you every success, and
Indeed.
thank you. And for the rest of us, well, I will get the link out. If you don't mind, I'll edit this and put it on our YouTube channel and send you a link so you can see what's there. You're welcome to use it, of course, because it's your content. So
that's fine. That's fine. No worry.
Thank
all
you
right.
very much indeed.
Thank you. Bye bye.
Thank you. Bye bye.
Bye bye.